Hello everyone, welcome back to This Is Not a Science channel, and today for our biology episode we will start on a new topic, a very important one indeed. It's about biological molecules. The reason why I say it's important because these are the building blocks of a lot of things in our life, especially ourselves, and it's all around in our diet, so it's very important for us to learn about these biological molecules. So let's start. First thing we will start off with is carbohydrates, and carbohydrates are probably a word that most people will be familiar with because it's all around our diet. In meal, you would either have carbohydrate in one way or another. So these include, for example, bread or sugar. Well, the word carbohydrate actually means carbon, hydrogen, and the ate at the end means oxygen. So basically molecules with C, H, and O. So these carbohydrates are examples of starch, sucrose, glucose, or glycogen. And in order to understand every single one of them, we need to know how they link together. We'll start with starch. Starch is probably the most common one. For example, bread contains starch, or rice contains starch. Well, starch is the largest form of carbohydrate. The reason why is because it coils up like this. So it's basically simple glucose molecules coiled up as a branch. Whereas glucose, on the other hand, they are the smallest starch unit. They're the smallest carbohydrate unit. And they look something like this. Well, starch is basically chains of glucose coiled up together in a compact form. And the reason why is because starch acts as a storage for plants, for example. So something like potato, that's a starch storage. So whenever plants make food from photosynthesis, they secrete it in the form of, they produce it in the form of glucose. Well, the thing with starch is, starch can easily convert to sucrose. Sucrose is basically a slightly smaller unit and not as small as glucose. And sucrose is used for transportation across the plant. And then they can convert sucrose into glucose, which is an easier form as an energy. On the other hand, you can also convert glucose back to sucrose. And you can convert sucrose back to starch as a unit for storage, like potatoes. And another form is glycogen. Well, glycogen is basically animal version of starch. It's how animals store their glucose. So instead of storing our glucose as starch, we store it as glycogen. And on the other hand, we can also convert it back into glucose. Next, we have lipids, or another word, fats and oil. So some examples are like cheese or oil, olive oil or animal oil, etc. So lipids, they are a very special molecule because they have a structure that looks really odd, like this. And it consists of one glycerol right here on the top, and we have 3 fatty acids. So this is how lipid structure look like. There is always one glycerol on the top, connected to 3 fatty acids. And this you have to remember because once we break down lipids, it's going to end up with 1 glycerol and 3 fatty acids. The next major one is proteins. Proteins are probably the most important molecules for us because it's what actually makes us grow or repair our tissue. And we can obtain protein from red meat, for example, or beans. So proteins are really important because they are chains like this. They are chains of amino acids. So why do we call it amino acids? Here, each and one of these individual molecules, they are amino acids. And there are 20 kinds of amino acids that human body needs. So it's very important that we have protein in our diet. So we can fix our tissues or make us grow, for example. And this is how an amino acid actually look like. The reason why we call it amino, because you see right here, there's a nitrogen here, we call it amino group. And we got a carboxyl group here that makes it an acid. And we have an R group, which is different for every single type of protein. So as I said before, we have 20 types of protein in our body. So there are 20 different types of R groups. And basically, the reason why this is so different from others is because, for example, like carbohydrate, it has CHO, carbon, hydrogen, and oxygen. Whereas amino acid, it has CHON, it has an extra nitrogen. And that's what's actually make it a protein, because it has this amino group right here. So these amino group will interact with each other and they form a sort of bond called peptide bond. And peptide bond are the black lines right here that holds the amino acid together. Amino acids then form a huge chain of primary amino acids. These then fold together to form helixes or sheaths, and then they fold all together to form a 3D structure or merge with other amino acid chains to form a final protein structure. So protein structures are actually really complex, and every single one of the amino acids are different. So even just one change in the amino acid sequence could lead to a completely different type of protein. According to the syllabus, there is a need for you to know how to test for every single one of these molecules, and these tests you have to memorize the procedures by heart. It's really important that we know these tests so we can identify what kind of food has carbohydrate, starch, protein, for example. First of all, the test that's the simplest is probably the iodine starch test. So what we do is, the procedure basically means we have to drop 2 to 3 drops of iodine solution on our sample. For example, our rice. So we drop 2 to 3 drops of iodine and we observe the changes. And the results are, basically, if the sample turns blue-black, it means that the sample contains starch. If there is absolutely no color change, that means it does not contain starch. And the reason why is because when we have starch, if you remember from the previous slides, starch has this sort of coiling shape. And the reason why is because iodine gets in between the coiling chains and therefore turn it into blue-black. So it looks something like this. If something contains starch, it's going to look blue-black. Whereas if it doesn't, it's just going to remain its original color of iodine solution, which is around yellow-brown. The next test is a bit different, and it's also a bit complicated because we are testing for reducing sugars, or basically just sugar in general. Well, it is to know that not every sugar are reducing, but the majority of them are. So we call this the Benedict test. What we do basically, the procedure is to drop 2 milliliters of Benedict solution in test tube sample. And then we have to put this sample with the Benedict solution in a 70-degree water bath for 3 to 5 minutes and wait for the color to change. The result is basically, if we see a dark red solution, it symbolizes a lot of reducing sugar. And if the solution did not change color, which maintains blue, we will categorize it as not much reducing sugar present. And the reason is because Benedict solution is basically copper sulfate, CuSO4. Well, the copper, once they got reduced, they show a sort of reddish color. So the reason why we call these reducing sugars are because these sugars have the ability to reduce copper. And once copper got reduced, the solution will appear red. So this is a chart for you to see. Once we have brick red, dark red precipitate, that basically means we have a lot of reducing sugar. If it's orange, we have slightly less, yellow, green, and therefore the least will be blue, where there is absolutely no reducing sugar. That means no color change at all, no copper got reduced. Next up, we have the Biuret protein test, and this one is people usually get this one and the Benedict test confused. Well, make sure you don't, because Biuret protein test, it's for protein. And the procedure is basically you have to add 2 milliliters of Biuret solution to a test tube of sample and leave it for 5 minutes. As simple as that. And you will observe the changes. Well, the results basically means if the solution turns purple, it means there are protein. If the solution did not change color, so remains blue, there is a negative. The test result is negative, which means there are no proteins. And the reason is because it turns purple because the Biuret solution actually detects peptide bonds. And if you remember what I said before from the amino acid chains, between the amino acid there is this bond holding the acids together, and that's what we call a peptide bond. So basically, Biuret solution, they are sensitive to peptide bonds, and once they detect peptide bond, they will turn purple. So it basically looks like this. If a solution contains protein, it's going to look purple like this. If it does not contain protein, it's going to look blue. And the final test will be the ethanol emulsion test, testing for lipids, fats, and oil. And how to do this test is actually pretty simple. All you have to do is just drop a few drops of ethanol and allow it to rest for around 1 to 2 minutes. Then you add distilled water. So distilled water actually means pure water, so nothing in it, just pure water, inside the test tube, and you will shake the test tube, basically mix it around, and you will observe the changes. Well, basically, the result will show if the test tube turns cloudy or sort of whitish, milkish color, then there are lipids present. If the test tube is still clear, that means there is no lipid at all. The reason is because fats and oil, they form something called micelles after contacting with water. You might have heard this phrasing as oil and water never mix. Well, that's it. Once you've added water, they wouldn't mix. So the white particles that you see here are actually micelles. That shows that this solution contains lipid, whereas when it doesn't, it's just going to remain clear. So that's it for today's video. I hope you've now learned the 4 tests that we need to know for the biological molecules, and it's important that you learn it because it will be tested on exams. It's probably the most vital part of this section of biology. So make sure you learn it and like the video, subscribe to my channel, and share it to all your friends who hate science. Thank you very much. See you next time.